Good afternoon, everybody. My name is Edward, working as Assistant Professor in the Department of Microbiology, St. Mary's College, Autonomous, Thuthukudi. Now I am going to discuss about the agglutination reactions and immunofluorescence method. To begin with, what is agglutination? Agglutination is an antigen-antibody reaction in which a particulate antigen combines with its antibody in the presence of electrolytes at a specified temperature and pH, resulting in the formation of visible clumping of particles. It occurs optimally when antigens and antibodies react in equivalent proportions. This reaction is analogous to the precipitation reaction in that antibodies act as a bridge to form a lattice network of antibodies and the cells that carry the antigen on their surface. Because cells are so much larger than a soluble antigen, the result is more visible when the cells aggregate into clumps. So as you see it in the diagram, the antigen and antibody, they combine together and make clumping. Now the step of agglutination. Agglutination generally involves in two steps, or it takes place in two steps. The first one is sensitization. It is a primary stage where binding of antigen and antibody takes place. Temperature, pH, ionic strength and incubation period influence the efficiency of sensitization or binding. Sensitization generally is not a visible reaction. Then lattice formation. It is a secondary stage where the antibody and multivalent antigen forms a stable network called lattice. A lattice exists a net-like configuration which consists of a stable network between sensitized antigen and antibody. It takes much time to occur than the sensitization. Lattice is a visible reaction. And type of agglutination test. There are generally two types of agglutination. One is direct agglutination, another one is indirect agglutination. Direct agglutination test is a reaction where antigens are found naturally on a particle, that is generally called as direct agglutination. Direct bacterial agglutination uses whole pathogens as a source of antigen. It measures the antibody level produced by a host infected with that pathogen. And the binding of antibodies to the surface antigens on the bacteria results in the visible clumps. And active agglutination can be of the following types. First one is slide or tile agglutination. Here the basic type of agglutination reaction is performed on a slide. That is why it is called a slide agglutination. And identification of bacterial types represent a classic example of slide agglutination. In this method, suspension of unknown antigen is kept on a slide and a drop of standardized antiserum is added, vice versa. A positive reaction is indicated by the formation of visible clump. The very best examples are Vidal test and RPR test. Then tube agglutination. It is an agglutination test performed in tube and standard quantitative technique for the determination of antibody titer. In this method, serum is added in a series of tubes and standard antigen suspensions are added to it. First of all, serum is diluted in the series of tubes and then standard antigens are suspended. After incubation, antigen-antibody reaction is indicated in the form of visible clumps. And third one is heterophile agglutination. This test depends on the demonstration of heterophilic antibodies in the serum that is present in certain bacterial infection. Antiglobulin test. The fourth one is antiglobulin test. This test is generally devised by Coombs, Moran and Race for the detection of incomplete Rh antibodies that generally do not agglutinate Rh positive erythrocyte in the saline. When serum containing incomplete anti-Rh antibodies is mixed with Rh positive erythrocyte in saline, The incomplete antibody antiglobulin coats the surface of erythrocyte but does not cause any agglutinin or any agglutination. When such erythrocytes are treated with antiglobulin or Coombs serum, then the cells are agglutinated, and the Coombs test can be direct as well as indirect. So this diagram explains about how the direct agglutination reaction taking place and indirect agglutination reaction. In the case of direct agglutination, particulate antigen corresponds with its antibody, binding with its corresponding antibody, then make visible agglomerate. In case of indirect agglutination, soluble antigen first bound to the sensitized particulate and then with corresponding antibody, finally it makes visible agglomerate. And this is the diagram explains about how the passive agglutination and reverse passive agglutination occurs. In case of passive agglutination, carrier molecule and soluble antigen, they make contact with the antibody and form agglutination in a passive manner. So in case of reverse passive agglutination, carrier molecule along with antibody that bind with the antigen, then it makes reverse passive agglutination. Now the indirect or passive agglutination. The passive agglutination generally employs carrier particles that are coated with soluble antigens. In this, either antibody or antigen is attached to certain intercarrier or inert carrier, or the cells that gets agglutinated when corresponding antigen or antibody reacts. This is an inert carrier, latex particles, carbon particles, or pandonite, and they are used as inert carriers. Example, antigens coated in latex particles, which is used in ASO test. When the antibody instead of antigen is adsorbed on the carrier particle for the detection of antigen, it is called reverse agglutination. Reverse agglutination can be of the following types: latex agglutination, heme agglutination, coagulation. Latex agglutination. The first one is latex agglutination. It employs the latex particle as a carrier of antigen or antibodies. In latex agglutination, many antibody and antigen molecules are bound to the latex beads, which increases the number of antigen binding sites. If corresponding antigen or antibody is present in a test serum, antigen-antibody bind and form visible cross-linked aggregates. Latex agglutination can also be performed with the antigen conjugated to the beads for testing the presence of antibodies in a serum specimen. In case of hemoagglutination or heme agglutination, RBCs are used as a carrier particles. RBCs of sheep, human, or chick can commonly be used. When RBCs are coated with antigen to detect antibodies in the serum, this test is called indirect heme agglutination test, or IHA. Heme agglutination uses erythrocytes as a biological carrier of bacterial antigen and purified polysaccharides or proteins for determining the presence of corresponding antibodies in a specimen. When antibodies are attached to the RBCs to detect microbial antigen, it is known as reverse passive agglutination. And finally, viral heme agglutination. Many viruses, including influenza, mumps, measles, that have the ability to agglutinate RBCs without antigen-antibody reaction. So this process is called viral heme agglutination. This heme agglutination can be inhibited by antibody specifically directed against the virus, and this phenomena is called heme agglutination inhibition. And advantage of this agglutination reaction, this reaction is very easy to perform, and the speed also very high, and it occurs only less than 10 minutes. The degree of sensitivity is very high, and at a time you can detect many different kinds of antibody, and with the exception of cold agglutinins, the tests are usually not affected by temperature condition. Applications. The applications are the following of this technique. The following are the applications of this technique. It is generally used in the clinical microbiology field in order to typing the recipient and donor blood and to detect the antibody presence and to quantify the presence of antibody in the patient blood. It also helps in serodiagnosis of toxoplasmosis. It helps in the determination of the RH factor. It is extensively used in techniques like latex hemagglutination. It helps to detect the type of antigen and quantify the amount of antigen present in the patient blood. And the next technique is immunofluorescence. What is immunofluorescence? To begin with, immunofluorescence is an assay which is used primarily on biological samples and it is classified or it is classically defined as a procedure to detect antigens in cellular context using antibodies. The specificity of antibodies to the antigen is the base for immunofluorescence. The property of certain dyes absorbing light rays at one particular wavelength and emitting them at different wavelength is known as fluorescence. So, immunofluorescence tests are also termed as fluorescence antibody. Fluorescent dyes such as fluorescein isothiocyanate and lysamine rhodamine can be tagged with antibody molecules. They emit blue-green and orange red fluorescence under UV light in the fluorescence microscope. It forms the basis of the immunological test. The tests are broadly classified into two types: direct immunofluorescence and indirect immunofluorescence. In case of direct immunofluorescence, it is used to detect unknown antigen in a cell or tissue by employing a known labeled antibody that interacts directly with the unknown antigen. If antigen is present, it reacts with the labeled antibody and the antibody coated antigen is observed under UV light of the fluorescence. It also involves use of labeled antiviral antibody. Indirect immunofluorescence test. Indirect immunofluorescence is a double antigen antibody technique. The unlabeled antibodies which are bound to the antigens are visualized by a fluorescent antiglobulin reagent detected at the unlabeled antigen. It involves in two steps. The first step is incubation of fixed antigen with unlabeled antibody. The second step is adding of second antibody to the first antigen antibody complex, which is labeled with fluorescent dye. And this diagram explains how the direct and indirect immunofluorescence work out. The first one is to the viral antigen, the labeled antibody is attached, and the indirect, in case of indirect, to the viral antigen, both labeled and unlabeled antibodies are combined. Advantage: the direct method of immunofluorescence have single labeled antiglobulin. It is a universal reagent. It is often more sensitive than the direct immunofluorescence. Applications: to detect specific antibodies in case of serodiagnosis of syphilis, leptospirosis. It also helps in identify the class of given antibody. It also helps in identifying and enumerating lymphocyte subpopulation. Then finally, to detect autoantibodies such as antinuclear antibodies in autoimmune diseases. That's all. This is the content related to immunofluorescence and agglutination.